Okay, hi everybody. Good afternoon. I myself, I'm a veterinarian, and before joining the industry some 20 years ago, I used to be a scientist working on actually on IBV and porcine, avian and porcine coronavirus in the in the UK. So let's now have a few minutes about veterinary coronaviruses and some links to COVID-19. A brief review about about all the some of the different technologies which are employed during the COVID epidemics, and some representing some major categories of vaccines, going from traditional like live attenuated vaccines to more to the most modern ones, to nucleic acid, RNA, DNA vaccines. If we start first with the live attenuated vaccines, these kind of vaccines replicate in the host. Normally, they do not cause disease, and sometimes there's some visible signs of vaccine virus replication. Overall, the efficacy of these kind of vaccines is quite good. They can be given as one dose most of the time, and the broad spectrum of protection. As an example from animal health, if you look at avian IBV vaccines, attenuated live vaccines, some like iBird, Marcel, iBros, representing a few of them, these category of vaccines have been kind of the workhorse in the last few decades, actually, to protect the poultry against IBV. These kind of vaccines are produced in eggs, and they also they are also attenuated in eggs to create the vaccine virus. Most modern approach recently applied as a COVID example is by Codogenics of making a similar category of vaccines, but instead of going through a lengthy and slow process of attenuation, this company used a computer-based algorithm to design an attenuated virus, and I think they are in phase 2, phase 3. Moving over to just an example on the right side of the screen, you see that just an example of how diverse IBV can be, many different serotypes and genotypes. And just to show that there are relatively few vaccines available around the world, or most importantly, the combination of these modified live vaccines available in the world have been very successful in protecting the poultry against all of these different variants of COVID-19 of IBV, which actually is much more diverse than COVID-19 itself. Moving over to inactivated vaccines, inactivated whole virus vaccines, most of the time they trigger antibody response in the vaccinated animals. In this case, the efficacy is a little bit antigen dependent. There are some better antigens. Sometimes there are not as good antigens in the vaccine. Pre-immunization most of the time is needed, or actually very beneficial, especially if I'm thinking of let's say an example of poultry again. The immunity can be long-term, and adjuvant is typically needed. In the animal health industry, I'm using the poultry again as an example. Most of the time, these vaccines are used as a booster once suitable live vaccines, live IBV vaccine in the case of coronavirus, are given to the birds. And then there can be inactivated monovalent vaccines like an IBK vaccines, but actually in real life, these vaccines are used in combination with 3 to 5 other antigens to give protections, for example, to the layers. In the case of COVID-19, actually the Chinese companies which are represented who are using this kind of technology, Sinopharm and Sinovac, for example, they use cell culture produced virus, inactivated, adjuvanted with aluminum hydroxide, and these kind of vaccines show they have a very good safety profile and reasonably good efficacy. Moving towards to subunit vaccine, which is already representing the more modern technology of vaccines. In this kind of vaccines, only the immunologically important part of the virus or the agent is produced. In this case, the spike we know very well by now, which again triggers a humoral immune response in the vaccinated animals. Efficacy can be antigen dependent. Sometimes you have better antigens, sometimes you have worse antigens. Immunity can be long-term, and adjuvant is needed in this case. Adjuvant is needed in this case. In the case of animal health, for example, an example for this kind of vaccines is porcine circovirus 2, PCV2, it's a subunit vaccine, or maybe better classified as a virus-like particle VLP vaccine produced, for example, in the baculovirus expression system. In the case of coronaviruses, it's very difficult to give you an example of which is a good example, a successful example of a subunit coronavirus vaccines, because despite lots of research, research programs, it's quite challenging and not yet successful to have these vaccines on the market. In the case of poultry, one reason is maybe the relatively high cost of producing such an antigen for the large number of poultry we have. In the case of humans, COVID-19, Novavax, the US company, is following this technology using a spike protein produced in bacuvirus system, adjuvanted, inducing strong immunity and good efficacy. Moving towards to viral vector vaccines, this is a very popular family. I mean, so as for the human treatments, so as for COVID-19, the same for veterinary applications. In this case, the immunologically important gene, the spike gene, is inserted into a harmless vector virus, which then being injected into the animal and produces the spike protein and induces actually both cellular immune response and also humoral immune response. The efficacy of these kind of vectors can depend on many things. I mean, vector vaccine and vector vaccine can be hugely different. A lot depends on the vector itself and the characteristics of the vector and the antigen itself, and including how the construct is made, what are the molecular details, promoters, and other minor details in the different constructs. 1 to 2 doses are typically needed depending on the construct of the vaccine, or mostly depending on the vector itself. They can induce long-term immunity, and in generally, adjuvants are not needed for this kind of vaccines. There's lots of examples from the animal health industry for these kind of vaccines, mostly using turkey herpes virus, a harmless vector which was never ever been pathogenic, HVT, and also fowlpox virus. When there are lots of vaccines which have been brought to the market actually since 1994, so over close to 30 years now, and most of them are still available and protecting chickens or billions of chickens over the world in many, many countries, in most cases against not only one, but sometimes 2 or maybe against 3 different diseases. In the human COVID field, this is also a very populated area or category of COVID vaccines. In this case, the companies are using adenovirus vectors. For example, chimpanzee adenovirus, human adenovirus 5 or 26 different serotypes, like AstraZeneca, Sputnik, and the Chinese companies, and Johnson and J&J as well. These vaccines are very efficacious, and sometimes the drawback of this kind of technology can be that there can be antibodies persisting even before vaccination, or can be induced against the vector itself, which can reduce the efficacy of the subsequent shots. However, a great benefit of, for example, the adenovirus vector technology is easy handling with stability and 4 degrees for weeks and months. Then finally, the nucleic acid or RNA vaccines. These are where the spike protein in this case is inserted into a synthetic RNA molecule, which is then packed into, let's say, lipid nanoparticle entering the cell, and actually the cells themselves which produce the antigen or the spike protein as well. Efficacy can depend a lot on the design of the RNA, on the antigen, and also the delivery tool, this LNP, the lipid nanoparticle, or maybe some other system. They play a very important role in the efficacy and also the amount of dose of this technology, of these vaccines as well. These vaccines have been very successful these days, as most of you know by now. In the Pfizer, BioNTech, Moderna vaccines representing this kind of technology, 2 doses, strong efficacy. In this case, there's no blocking effect is known against these kind of vectors. In the animal health industry, these vectors are also under development in research phase in several companies. And I would say, unfortunately, the veterinary health doesn't have a crisis situation today, and there's no expedited speed up registration procedure. So it will take a bit longer to bring these new technology vaccines to the market compared to how successful it was in the case of COVID-19, when actually within a year, these kind of vaccines were already available to humans for vaccination. And finally, maybe a strange question. I mean, are we lucky or are we not lucky to have a SARS-CoV-2 as a pandemic? I mean, it can be a strange question, but is it a good virus or not a good virus to have this as a pandemic? I mean, of course, it would be better to have nothing. But if we have a pandemic already, maybe let's say this is quite fortunate to have this virus. I mean, just looking at the history, all this huge amount of knowledge which has been built over the last, I think, more than 40 years now, starting with Béla Lomnici, a Hungarian veterinarian who was the first one to describe the genome of infectious bronchitis virus, followed by Dave Kavanagh in the UK, who was the first one to describe and characterize the spike protein, the protein we hear so much about these days, followed up with another British group of scientists, Bursnell et al., who completed the first sequence of, again, IBV as a type strain of coronaviruses in 1987. Then you had to wait 13 years up to 2000, when a group of scientists led by Luis and Juanas in Madrid was the first in the world actually to create an infectious clone, an infectious bacterial artificial chromosome, and created the first man-made coronavirus, which in this case was a TGV infectious clone. Then again, a few years ago, just an example, Gerd Sutter in Germany was successfully constructed a vector vaccine against MERS based on Vaccinia MVA virus, and has given a very good candidate, which then eventually the vaccine was not registered because the disease kind of became non-significant later on. So thank you very much for your attention.